This video is about anaplerosis, the creation of citric acid cycle intermediates. Running the cycle requires a pool of intermediates, and although the intermediates are interconverted rapidly during the cycle, there is no net breakdown or synthesis of the total pool of intermediates. However, there are circumstances in which the intermediates need to be created, and this is the process of anaplerosis. Now, because the intermediates are interconverted rapidly, making any one intermediate is sufficient for anaplerosis. So the creation of citrate or oxaloacetate or alpha-ketoglutarate are all equivalent. It's important to distinguish between open and closed citric acid cycles. Closed cycles involve simply interconverting intermediates. Acetate comes in, carbon dioxide comes out, and the intermediates go around the cycle, but intermediates don't enter or leave. Most tissues do this. So in brain, for example, the citric acid cycle runs primarily for energy production, and the need for anaplerosis is low. In tissues with open cycles, intermediates leave the cycle because they're used by other pathways. And in this case, the demand for anaplerosis is much higher. An open cycle, or the places where open cycles occur, is shown on this slide. So some examples include the synthesis of fatty acids, which are made by citrate. This occurs in the liver. The synthesis of amino acids, so glutamate can be made from alpha-ketoglutarate. Heme is made from succinyl-CoA. And gluconeogenesis in the liver and kidney uses either malate or oxaloacetate. And so in that case, these intermediates leave the pathway. Now, it's important to note that all of these biosynthetic processes are under strong regulation, so that even tissues that have open cycles only have them under appropriate metabolic conditions. And so a good example is liver, which will remove malate for gluconeogenesis in the fasted state, but will remove citrate for fatty acid synthesis in the fed state. So even though both of these examples of an open cycle occur, they never occur in the liver at the same time. There are a number of reactions that are anaplerotic that can make citric acid cycle intermediates, and we're now going to talk about those. The first and most important is pyruvatecarboxylase. This occurs in all tissues, so every tissue has some need for anaplerosis, and pyruvatecarboxylase is the enzyme that does it. It turns pyruvate into oxaloacetate. And important things to note is that this is an energy-requiring reaction. ATP is hydrolyzed. The extra carbon for turning a 3-carbon pyruvate into a 4-carbon oxaloacetate comes from bicarbonate. And as in most carboxylation reactions in mammals, biotin, the vitamin, is the CO2 carrier. This reaction is regulated. It has acetyl-CoA as a positive allosteric activator. So when acetyl-CoA piles up inside the mitochondria, it's a signal that it's time to run the citric acid cycle faster, stimulates anaplerosis with the production of more oxaloacetate. The other anaplerotic reactions tend to occur only in tissues that have an open cycle. So we're going to talk about those now. The next ones involve anaplerosis from amino acids. So the first of these makes alpha-ketoglutarate from the amino acid glutamate. The enzyme that does it is glutamate dehydrogenase. This enzyme is usually expressed only in the liver, can also be expressed in the kidney during prolonged fasting, and it has vitamin B6 as a cofactor. Now, this enzyme serves two functions. One is that it's anaplerotic in liver, and the other is, you'll recall, that it produces ammonia for the urea cycle, for the synthesis of urea, and that explains why this enzyme is liver only. Half of the nitrogens for the urea cycle come from ammonia, and much of this ammonia is produced by this enzyme. This enzyme has allosteric regulators. It's activated by high supply of the amino acid leucine, and I've never seen a good explanation for the regulation of this enzyme by nucleotides. Now, amino acids can also be anaplerotic in a more complicated way. Glutamate dehydrogenase is the only enzyme that can remove nitrogens directly as ammonia, but by combining with transaminases, other amino acids can also participate in anaplerosis. So here's an example. Here's the enzyme AST. which takes aspartate and alpha-ketoglutarate, transfers the nitrogen from aspartate to alpha-ketoglutarate. The products are then oxaloacetate and glutamate. This reaction by itself is not anaplerotic because you start with one citric acid cycle intermediate and you end up with another. There's no net production. However, this enzyme or this reaction can combine with glutamate dehydrogenase with the result that the glutamate that's produced in this reaction is then turned into alpha-ketoglutarate. And so the result is, if you sum this reaction and this reaction, the net reaction is the conversion of aspartate to oxaloacetate, reduction of NAD, and the production of ammonia. And so as a consequence, aspartate aminotransferase combined with glutamate dehydrogenase is anaplerotic. The next anaplerotic reaction is capable of producing succinyl-CoA, and the starting material are these amino acids, and it can also use propionyl-CoA, which is produced from the breakdown of these amino acids, but also from the oxidation of some fatty acids. There's a short pathway that's shown on this slide. Turns propionyl-CoA into methylmalonyl-CoA. This is a carboxylation reaction that's very similar to pyruvatecarboxylase. It puts on an extra carbon from bicarbonate with the production of this compound, methylmalonyl-CoA. Then methylmalonyl-CoA undergoes a racemization, so it changes, the optical isomers are switched. And finally, there's a migration in which this carbon moves from carbon 2 to carbon 3 with the production of succinyl-CoA. And this reaction has a vitamin B12 as a cofactor. Vitamin B12 is used in exactly 2 reactions in humans, and this is one of them. And yet, quite remarkably, it's absolutely required for human health. Now, the last anaplerotic reaction that we need to think about is the purine nucleotide cycle. This occurs in skeletal muscle. Here's the purine nucleotide cycle. It starts with a molecule of AMP, which is deaminated, loss of ammonia to form IMP. IMP then is condensed with a molecule of aspartate. Hydrolysis of GTP is required to drive this. And this adduct is then split at this bond to to regenerate AMP with the release of fumarate. So the net reaction is the hydrolysis of GTP and the conversion of aspartate to a molecule of ammonia and fumarate. So it's turning this amino acid into a citric acid cycle intermediate. As I said, this reaction is important both during exercise. During strenuous exercise, there's an increase in the size of the pool of the citric acid cycle in skeletal muscle. The purine nucleotide cycle does it. And in addition, it's important during prolonged fasting when there's net breakdown. So those are the important anaplerotic reactions. And just to sum up what's in this video, we've talked about why anaplerosis is necessary and about the different requirements in cells with open and closed cycles. It's good to keep in mind that in a fasting individual, gluconeogenesis can be making 100 to 150 grams per day of glucose, and that glucose is coming largely from citric acid cycle intermediate, so there's a strong demand for anaplerosis. And then we talked about the individual reactions, with pyruvatecarboxylase being the single most widely distributed, and the others being important mostly in liver and muscle tissues that have open citric acid cycles.